Well, good evening, everybody. For those who don't know me, I'm Aberrant. I'm the Executive Dean of the Faculty of Health and Medical Sciences. I'd like to start by acknowledging the Ghana people, the original custodians of the Adelaide Plains, and the land on which the university's campuses here, Roseworthy, Waite and Theberton are built. So I'm delighted to welcome you to the annual Florey Public Lecture. Thank you for joining us to this very important occasion. The annual Florey Lecture is hosted by the Florey Research Foundation, and each year the Foundation invites a highly distinguished health sciences researcher to Adelaide. And this year I'm delighted to introduce this year's Florey lecturer, Professor Jane Visvader. So Jane is a scientist specialising in breast cancer research, working in the Walter and Eliza Hall Institute in Melbourne, the WEHI, where she's the joint head of the breast cancer laboratory with Professor G Linderman. Jane carried out her PhD studies here at the University of Adelaide and subsequently held positions as a postdoctoral fellow at the Salk Institute in San Diego and then subsequently in the Children's Hospital and Harvard Medical School in Boston. And in 1998, she made the transition to mammary gland development and breast cancer research with an appointment to the Victorian Breast Cancer Research Consortium as group leader based at the WEHI. She is a named inventor on 5 patents relating to cancer research focused on stem cell isolation and diagnostics. She's a fellow of the Australian Academy of Science and earlier this year was inducted as a fellow of the Australian Academy of Health and Medical Sciences. She has received a string of prestigious awards, including the Shearer Sifting Lectureship from the German Cancer Centre, the GlaxoSmithKline Award for Research Excellence, the Royal Society of Victoria Medal for Excellence in Research, and earlier this year, the Lemberg Medal for Excellence in Biochemistry and Molecular Biology. Over the past decade, Jane and her team have made important contributions to the fields of mammary biology and breast cancer by isolating stem cells, defining master regulators of mammary gland development, and identifying genetic lesions that drive oncogenesis. So without further ado, I'd like to introduce Jane to discuss mapping the mammary gland to understand breast cancer. Jane.
Thank you very much for that lovely introduction. I'd firstly like to thank the Foundation for the very kind invitation to deliver this lecture. I'm very honoured. It's also always a pleasure to come back to Adelaide, and actually I've seen some faces tonight. They're my old Bachelor of Science friends, very friendly faces. I'm feeling quite nervous now because I haven't seen them in such a long time. It really was lovely. It's lovely. All right, so let's turn to the topic for this evening, breast cancer. It probably needs very little introduction, and what I'm going to try and do in this talk is take you on a journey through the last 10 years of our research. I hope it's not too complicated. If it is, please feel free to ask me questions at the end. So breast cancer is the most frequent cancer to affect women. And, sorry, I've already made a mistake. And this is actually the MCG with about 13,500 women in peak ponchos in the month of October to help raise funds for breast cancer research. So, as mentioned, it is the most common cancer to afflict women. It accounts for almost a third of these cancers, as you can see by the red, here we are, the red pie chart. Now, despite significant strides that have been made over the last 20 years, it does remain a significant problem. So we've seen a huge decrease in mortality over the last 20 years, largely due to adjuvant therapy and mammographic screening. Nevertheless, there is a gap, and about 515,000 women die each year worldwide due to breast cancer, highlighting the significance of this problem. So breast cancer actually arises in the epithelial tissue. So breast comprises a ductal tree comprised of epithelial cells that's embedded in a fatty stromal matrix. And the ends of the ducts constitute these TDLUs. They are called terminal ductal lobular alveolar units. And this is actually where the breast cancers normally arise. So in these, within the ends of the ducts, as you can see here. So it's very dense epithelial tissue. That's a cross-section through one of them. And breast cancer is not a single disease. It is highly heterogeneous. Pathologists say that there are approximately 18 different subtypes, some of which are shown here. So visually, one can appreciate just how diverse breast cancer can be at a histological level. Most, of course, fall into the invasive ductal carcinoma subgroup. At a molecular level, so that's looking at all the genes that are either expressed or are switched off in a particular type of cancer, you can see that there is tremendous heterogeneity. So the green and the red represent genes that are on and off, and it means that there are distinct subtypes at the molecular level. And in fact, although this is really complicated because they say there are about 10, but actually I think they can be stratified into 5 major subtypes: the luminal A, the luminal B, the HER2 or ERB2 overexpressing, basal-like, these are the most aggressive, and the claudin lows. So there are 5 major molecular subtypes of breast cancer. So this brings me really to the tenet behind our research. We are trying to understand the molecular and cellular mechanisms that underlie breast cancer, and we are doing this through the prism of normal development and actually identifying the normal epithelial cell types that live in breast tissue. And so this is where my talk will start, and then I'll come back to breast cancer. So the mammary gland, or breast, is a very dynamic organ. And it undergoes cycles of cell growth, differentiation, and cell death, or apoptosis, with every cycle of pregnancy, lactation, and regression. So it's constantly undergoing these cycles, and of course we have to use animal models necessarily for most of our research. And this cycling is all under the influence of ovarian hormones, and this will be a theme that I will come back to. Well, when I said that we're trying to understand mechanisms underlying breast cancer through the prism of development, I truly mean it, and this was made possible by a very talented French postdoc, Anne Rios. When she came to our lab 5 years ago, she opened up a new way of visualizing the mammary gland, which had not been done before. So this represents a rudimentary ductal tree before puberty in a mouse. So you can see a very rudimentary ductal tree compared to the tree that I showed you on the earlier slide for human breast tissue. So we are switching to mouse until the very end. And when so we'll start with this rudimentary tree, which is present. This is just to orient you, this is the nipple region, and this is called the fat pad. So this tree is very basic at the beginning of puberty. But during puberty, we see elaboration of the tree into a beautiful branched network, as can be seen here. So this whole process is driven by bulbous structures, these yellow bulbous structures that lie at the ends of these tiny branches. They are the structures that drive elongation and branching through the fat pad until they reach the periphery of the fat pad, and then ductal growth ceases. So it's really a very beautiful system, and this happens in a directional fashion. It can't go backwards. It happens in a very strict and directional fashion. Now here we're going to take a short movie which highlights the blue cells of the inner luminal layer and the outer layer of green cells of the myoepithelial cells. So there are 2 layers that constitute the ducts: an inner layer of the cuboidal cells and an outer layer of highly elongated cells here. And it wasn't, you can see just how elongated they are. It wasn't until Anne performed this type of analysis that we understood that myoepithelial cells could be up to 100 microns or so. So the next movie I hope you can appreciate the power of this type of 3D confocal imaging. So we're moving through a duct now. You can see this, you saw the cells lining the duct, and now we see the surface of the duct together with the blood vessels and surrounding parenchyma. So this is a very powerful way of visualizing what is happening in real tissue rather than fixed tissue. So that's the inside of the duct. So actually one 3D image that I just showed you represents approximately 50,000 standard 2D images. And when Anne was studying the lactating mammary gland, she came across a very surprising finding, which is going to be highlighted in the next movie. So she was trying to, it's actually quite slow, but we're going to move through the lactating mammary gland. This is the duct. And these are the grape-like alveolar units that produce milk. So these are the milk-producing units. And so you can see a whole cluster of these lobular units. And now we are going to zoom in and take an optical section through one of these. And you can see the surprise that she found was that most of the cells contained 2 nuclei. So the field, this was a complete surprise, because before that time we thought that there was just 1 nucleus. So Anne and Nyang went on to show that it is absolutely essential to have cells with 2 nuclei for functional milk production. And if you actually interfere with this process and stop the binucleation, then there's very little milk and lactation fails. And this is conserved across at least 5 mammalian species, so it's a very intriguing finding. So now I'm going to go back, and this is really going to be the central focus of the talk. That was just a snippet. I'm going to go back to 2006, when our lab, together with Connie Eve's lab, isolated the mouse mammary stem cell. So these were fractionated. There were 4 populations, but the stem cells only reside in this population here. And even a single cell that is sorted from this population and then implanted into a fat pad, even a single cell was capable of regenerating the entire ductal tree. And this work was carried out by Marc Shackleton and Francois Vaillant. And it was an incredibly challenging project. So importantly, we tried to adapt the same methodology to human breast tissue. And this is a 3D culture. It's actually the pearl tiara that Elgene grew from a single stem cell. So you can see that it's trying to form a duct in this 3D culture system. And when these single cell suspensions were transplanted into mouse fat pads, for obvious reasons, you can see that they too were capable of giving rise to ductal structures with an inner luminal layer and an outer layer of myepithelial cells. So putting this together, this is the emerging picture of the hierarchy, but I should add that it's really, it might look complicated, but this is the most simplified version of the hierarchy. So there is a stem cell, an adult stem cell that resides in breast tissue. It sits at the apex of the hierarchy, and it gives rise to the 3 mature subtypes that are found in breast tissue: the ductal cells, the alveolar or milk-producing cells, and the elongated myepithelial cells. So this cell gives rise to these cells by virtue of a series of daughter cells, which are called progenitors. And Mara-Elias in our lab spent years isolating and characterizing the luminal progenitor and the alveolar progenitor. Now really, the most important thing here is not to take note of all the arrows and everything, but to appreciate that this setup is functionally conserved between mouse and human breast tissue, and that if you look at the gene expression or molecular profiles of certain cells across species, mouse and human, they are highly conserved. So it validates the use of animal models for, or especially the mouse, for our type of work. Now, over the last 10 years, our lab has gone to enormous lengths to try and identify molecular regulators. And these are just some of them, so that we try to work out which genes are important in controlling which cells and which processes. But I think one of the most interesting still to me is GATA3. This is a gene that is very important in luminal A cancers. The reason why luminal A breast cancers have a pretty good prognosis is because they have high levels of this gene. This gene promotes maturation of a progenitor or an immature cell to a differentiated cell, and that is why these cancers proliferate less. They're more mature. And I'm certain, based on a number of findings from our lab and others, that this gene is the key. So I wanted to go over some of our recent data because the stem cell itself has come into question. It's actually a highly controversial area in the field because the stem cell was shown by a transplantation assay, but the question is, can you actually track this cell in vivo, in live tissue, or tissue that is freshly isolated from the mammary gland? And so in order to do this, we needed to embark on combining 3D imaging with, which is very high resolution, as I showed you, with an in vivo lineage tracing method. And this is really very topical in the field. And this whole methodology relies on using a lineage-specific promoter upstream of a reverse TET transactivator. It's using the whole TET operon system from bacteria. So you have the TET operon upstream of the Cre recombinase, and then you have to make a triple transgenic strain, and you need to have a fluorescent protein. So in this case we used a tomato. It's a highly fluorescent protein. And so all of the cells in which this promoter is active, so will carry out Cre recombination and turn on expression of the fluorescent reporter. And those cells will be permanently marked for their entire lifespan. The other reporter locus that we used, not only the tomato, but this beautiful multicolored confetti locus, where Cre-mediated recombination leads to stochastic activation of a single color, either green, yellow, red, or blue, and it allows a clonal analysis. And I'll show you what I mean by that in a few slides. So we use two different reporters. Now, to mark the luminal lineage, so remember there are two lineages. There is the luminal, which comprises both the ductal and the alveolar cells, and then we've got the Maya epithelial lineage, so these highly elongated ones. To mark the luminal lineage, we used L5, which is a beautiful defining marker of cells, these daughter cells in both mouse and human. To mark the basal lineage, we chose keratin 5, and because there were very few other markers available at the time. Still, it's a big problem in the field. There are no highly specific markers of these stem cells. But the rationale behind this was that if keratin 5 marked a bipotential stem cell that could give rise to two lineages, then all cells within the mammary gland would be labelled, or a substantial proportion. So we delivered a pulse of doxycycline to activate the whole reverse TET transactivator TET operon system, and it was either delivered at the onset of puberty or in adulthood. And just to remind you, this leads to indelible marking of the cells and their progeny for their entire lifespan. So L5 was indeed shown to be highly specific for these cuboidal luminal cells as shown here, and most importantly, FACS analysis verified that it was luminal specific. And this was a 125 kb construct. And when the triple transgenic system was incorporated with the confetti locus, such that only one colour would be activated in an individual cell, you can see a beautiful sea of colour here in the terminal end bud and also in the ducts. And this told us that multiple progenitor cells are important for driving morphogenesis and growth of the mammary gland. So that's the whole elongation process in puberty. We also showed that it was important for their maintenance. Now moving to the other lineage, so those highly elongated cells, we use this keratin 5 promoter, and as you can see, we could verify that the model that was generated is highly specific. Only the outer layer of cells were labelled. Also, most importantly, again, seen by fax analysis, because this is the only way to truly quantitate what is happening at a global level. Now again, we saw a sea of colour. So the 4 colours, and they were stochastically activated. They occurred in a 1-to-1-to-1 ratio. And if you looked a little closer, here you can see these elongated cells and more cuboidal cells. So here we were starting to see 2 different morphologies. But to truly address the question of whether one cell could give rise to cells in both lineages, both the luminal and the myoepithelial, it was necessary to track the fate of these K5 marked cells at a clonal level. So on a very short chase period, you can see that there are small clones, and these clones were enumerated by Anne, actually at the microscope from both the inside and the outside of the ducts. Here's a typical clone with 1 elongated myoepithelial cell and a few luminal cells. I think 7 was a very typical clone that she saw. And this proved that one marked cell, could indeed give rise to luminal and myoepithelial cells. So it proves that stem cells can have bipotential character. And interestingly, and perhaps not surprisingly, we saw expansion of these clonal regions after a longer chase. So you can see a big red domain and a YFP domain there. With the fax analysis, these labelled cells fell into both the luminal compartment and the basal compartment, but perhaps more interestingly, we could show quantitatively that there's a huge expansion of these luminal cells between 2 days after the doxycycline and 8 weeks after the doxycycline. So it really proves unequivocally that stem cells exist in mammary tissue or breast tissue. So what about pregnancy and involution that occurs after the cessation of lactation? Well, here we see that many stem cells must be recruited in order to drive the formation of these lobular alveolar units which produce milk by the end of lactation, by the end of pregnancy rather. So you can see that many cells have been recruited here, implying that these cells can give rise to every cell type in breast tissue, not just the ductal cells and the myoepithelial cells, but also the milk-producing units. And finally, they are also actively recruited during the remodelling process, because when lactation finishes, it's important for all the lobular alveolar units to remodel. The whole gland has to restructure to a pre-pregnant state. And it seems clear that these stem cells must be recruited by virtue of the colour. And you can see the cuboidal cells and the highly elongated myoepithelial cells. Again, they have bipotential character. This was an important finding because after a 20-week chase, and Anne has now done a 2-year chase, it was clear that the ducts comprised 1 or 2 colours. So they weren't multicoloured anymore. They either had 1 or 2 colours. This is an example of a YFP and RFP coloured duct. And it implies that very few stem cells are involved in maintaining the integrity of an individual branch or duct. But that this process is very active. You can see the whole duct is labelled, which means that the tissue must be turning over at a very reasonable rate. And if you take sections, you can see the elongated cells and the cuboidal cells. So these are like clonal patches comprising both cell types. So in summary, we can trace this cell by this method of lineage tracing, and we can also demonstrate its existence by the transplantation assay. So it was very important to use both strategies. The luminal progenitor actually has been proven by both strategies as well. So I think there is no question that this cell exists, and this cell exists also in the live tissue. And if we just talk about the salient features of this work, we think that these mammary stem cells and daughter cells are very important for all of these processes. So they're important for morphogenesis during puberty, they're important for for maintenance of the ducts in the adult. They're important for the formation of these alveolar units during pregnancy, and of course this is absolutely required for lactation. However, when it comes to involution, we could show that only the stem cells are important, not these daughter cells. So that distinguishes stem cells from the other cells that lie downstream. So unipotent stem and progenitor cells are very important workhorses, but stem cells have a major role in long-term homeostasis, as I showed you with the yellow and red coloured duct, and in remodelling during involution. And the fact their longevity suggests that they are prime targets of mutagenesis, and I think we see a very clear example of this with the teenage girls during Hiroshima, I should say, Hiroshima. Okay, so now I'm going to move on to the second part of the talk, which is trying to understand the impact of ovarian hormones on the hierarchy. And it has been known for decades that these pink cells that express the estrogen receptor and progesterone receptor are intercalated with cells, the pale pink ones, that do not express these receptors, yet these pale pink cells are the ones that are turning over and proliferating, whereas these hormone receptor positive ones are more sedentary. They sit there, they are not highly proliferating. So understanding what the mechanisms at play here obviously is very important, because there are decades of evidence showing that prolonged hormone exposure to ovarian hormones increases breast cancer risk. There is absolutely flawless evidence for this. So we wanted to understand the impact of ovarian hormones on this hierarchy, which we are so focused on understanding. And you'll see, because it has so many implications for understanding the cell types that are predisposed to becoming cancerous. So I guess it was 2007, Murray showed that both mouse and human mammary stem cells lack the estrogen and progesterone receptors. So this cell does not express the hormone receptors. Yet we were able to show that these stem cells are exquisitely sensitive to these hormones. And as an example, if you overectomize an animal and transplant their stem cells into another animal, you can see that not only do we see a huge decrease in the number of stem cells, but also their ability to repopulate the fat pad is severely compromised. So mammary stem cells are highly sensitive to female ovarian hormone deprivation. Conversely, if we turn to pregnancy, rather than a decrease, we see an increase in this population where the stem cells live. And importantly, if we assay for the function of stem cells by using a transplantation assay, this is a very important readout of stem cell activity, there is an 11-fold increase in the number of active stem cells. So this augmented mammary stem cell pool presumably is responsible for driving the milk-producing alveolar cell expansion that accompanies pregnancy. So this contrasts with the ovarectomy situation. I think I just want to draw your attention only to the headline. So the part of the mechanism here is going to involve the epigenome, in particular the repressive K27 methylation mark, which decorates the histones and helps to dictate whether a gene is turned on or off. But certainly we found that the epigenome is very important ultimately at regulating gene expression under in different hormonal environments. So the next obvious question to answer was, well, we've got stem cells that don't express the hormone receptors, yet they are highly responsive to hormones. So how does this happen? It's called a paracrine mechanism, and it necessarily involves factors that are produced by one cell that are then secreted and influence the cell that does not express the hormone receptors themselves. And so to cut a long story short, Bhupinder and Murray carried out a lot of gene expression analyses, and they identified a particular gene called rank ligand. And through a series of in vivo and in vitro experiments, rank ligand was shown to be a key paracrine mediator of hormonal signaling to stem cells. And importantly, other groups in the area have shown that it is a progesterone target itself. So this is one key player. And just to summarise how this is operating, ovarian hormones will act on this ductal cell that expresses the receptors for the ovarian hormones. And, for example, under a progesterone stimulus, this will lead to the production and secretion of rank ligand, which then binds to its receptor, rank, which is expressed on the stem cell itself. And then this actually orchestrates expansion, which we saw in pregnancy. Remember, we saw a 10-fold expansion of the stem cell population. So this is at least one mechanism at play. So just to reiterate, hormone deprivation and anti-estrogens decrease the mammary stem cell pool and their activity. Excess hormones, either injected or under the pregnancy milieu, increase mammary stem cell function. Aging is actually associated with an increased mammary stem cell pool. Pregnancy dramatically increased the size and function of mammary stem cells. And rank ligand is a key factor that is very important for governing the proliferation of breast epithelial cells. So how can this knowledge be applied to tackling breast cancer? So this brings me to the last part of the talk, which is concerned with using the hierarchy to identify cells of origin of cancer and ultimately the identification of a targetable pathway for prevention. So we come back to one of these first slides that I showed, which depicts the molecular heterogeneity and the 5 major subtypes of breast cancer. A key question in the field, and it's still yet to be answered, is do these different subtypes of breast cancer reflect a different cell of origin? So what do I mean by that? So there are two main hypotheses that have been put forward to explain intertumoral heterogeneity. There's the genetic mutation model, which posits that different genetic errors, whether it's upregulation or downregulation of a specific gene or pathway, different oncogenic events lead to different subtypes of breast cancer or cancer. And the second model, but it's not mutually exclusive, is the cell of origin model, which postulates that even the same oncogenic event, AAA, in different cell types that lie along the hierarchy of cells that is found in every tissue of our body, will ultimately result in a different subtype of cancer. So this is going to be the focus of the next part. And to get a handle on this, we initially took the gene signatures that we derived from highly purified populations for the mammary stem cell enriched, the daughter cells and the mature luminal cells. So these gene signatures of the genes that were on and off were delineated. And they were used to interrogate the different clusters or subtypes of breast cancer. And the result was a very striking correlation between the gene expression signature of this stem cell and the Claudin-Low subgroup of breast cancer. And this was a bit surprising, but I won't go into that. But the biggest surprise was that the signature of the luminal progenitor is most concordant with that of the basal subtype of breast cancer. And not surprisingly, the ductal cell had a signature that was very close to that of the luminal A, the best prognosis group of breast cancer. So that was not surprising, but the rest was. So to explore this a little further, to really understand the relationship between normal cell types and those that are predisposed to cancer and ultimately give rise to different subtypes of breast cancer, we turn to BRCA1 mutation carriers. And this is because these women usually develop very aggressive cancers. They are quite often at a young age, late 20s, 30s, and this basal-like phenotype means that they do not express the estrogen receptor or progesterone receptor, so they cannot be given endocrine therapy. They can't be given tamoxifen, aromatase inhibitors. There are very few therapeutic options available for these women besides chemo. So it's a very aggressive subtype, and these women have, there's about a 70% chance of developing breast cancer before the age of 70. This also applies to a second breast cancer, and they're highly susceptible to ovarian cancer as well. But we had a very unique resource here because we were very grateful to these women and through their provision of tissue in the precancerous phase. So this is well before there's any tumour. They undergo a prophylactic mastectomy and have kindly donated their tissue to the laboratory, and we were through this type of process we were able to see that the luminal progenitor population here was expanded compared to non-carriers, so women undergoing reduction mammoplasties. So actually it had long been thought in the whole mammary gland and breast cancer field that this is the cell that would have been susceptible to becoming cancerous in BRCA1 mutation carriers. But actually, if anything, you can see that it's diminished. You can see there are far fewer cells. So the perturbation was restricted to this luminal progenitor population, not the basal subset. And importantly, these cells were aberrant, so they could grow they could grow in 3D assays in the absence of growth factors, whereas their normal counterparts required these 27 growth factors. So they were clearly perturbed. They were not normal cells. And their gene signature, so the gene signature taken from these BRCA1 mutation carriers in the precancerous phase, so I just want to really reiterate that, was very similar to that of a normal luminal progenitor signature. So putting all of that together, it's not only our work but also two subsequent papers point to this cell as the one that goes awry in breast cancer patient, in BRCA1 mutation carriers. So this cell is expanded, it has aberrant properties, and it almost certainly is the cell type that gives rise to these aggressive basal-like cancers. So this brings me to rank, because the critical question here is, well, it's important to identify the cell type that seems to be prone to becoming cancerous in BRCA1 mutation carriers, but how can we use this information to improve the outcome for these women and to actually prevent breast cancer as the ultimate goal? So this brings me to rank. So rank is incredibly important for bone or osteoclast formation and activation. As I mentioned before, rank ligand is a key paracrine effector of progesterone action. So I showed that in the mouse models. And we know that rank ligand and hormonal signaling are deregulated in BRCA1 mutation carriers. So we needed a very strong rationale to pursue this avenue of research. But when we went through the literature, there were so many different facets pointing to a role for rank ligand in these BRCA1 mutation carriers. We also know from the work of Penninger and others that inhibition of the rank receptor rank ligand signaling axis can delay tumorigenesis. So we had a strong rationale for addressing a role for this rank receptor, which is expressed on the surface of cells as a potential therapeutic target. So all of this work was carried out by an incredibly talented PhD student, Emma Nolan. So if we look at normal breast tissue from women, you can see that only the luminal progenitor, but not the basal or myepithelial cells, a subset express rank. But this population is expanded. It's expanded in tissue taken from BRCA1 mutation carriers. And you can appreciate that more over here. It's quantified over here. So we see no expression in the basal compartment. It's restricted to the luminal progenitor or daughter cell, and it is overtly expanded in these women. Moreover, when these cells were assayed for their growth properties, one can see that the rank positive cells are far more proliferative. They gave rise to much bigger and many more colonies than rank negative cells in the same luminal progenitor population. And if you compare the rank positive cells to their wild type counterparts, so these women do not carry a BRCA1 or BRCA2 mutation, you can also see that these cells are far more proliferative. Not only are they more proliferative, but they are more susceptible to accumulating DNA damage. And you can see this here. These are comet tails, which represent, so the length of the comet tail is a reflection of the number of single-stranded and double-stranded breaks. And you can see that it is this rank-positive population that has much longer comet tails in response to irradiation or carcinogen hydroxyurea, and this is untreated. But even in the untreated case, these rank-positive cells have a basal level of DNA damage, and so it points to an intrinsic defect in their DNA repair pathways. And that is consistent with, of course, BRCA1 being a very important tumour suppressor gene that has a role in homologous recombination. That's a very critical DNA repair pathway. So all of this put together strongly implicates rank-positive cells as prime targets for transformation in breast tissue. And so we know that from the work of others that progesterone in blood, so serum levels of progesterone can be elevated in BRCA1 mutation carriers. And so this is sending a signal by virtue of rank ligand to the rank-positive luminal progenitor cell, and this population is expanded and hyperproliferative in the tissue from BRCA1 mutation carriers. It's also highly predisposed to acquiring genetic mutations, so the end result is a cell that is undergoing these genetic hits and finally giving rise to a basal-like tumour. So it led to the obvious question, given all of this information, is rank ligand a potential therapeutic target for breast cancer prevention in BRCA1 mutation carriers? And BRCA1 has now really become known as the Angelina Jolie gene. So Emma used two different approaches here. So she firstly grew organoids, so these are human breast organoids, ex vivo, so in a tissue culture lab. And it was very important to keep the fragments intact, because if the cells were prized apart, we would lose the paracrine signaling, because remember that we've got these hormone receptor positive cells signaling to hormone receptor negative cells, so they need these paracrine signals to be retained. So she developed this assay, and it was, you can see the integrity of the layers is beautifully intact. Importantly, she showed that when she made organoids from BRCA1 mutation tissue, so it's heterozygous for this mutation, that these cells were hyper-responsive to progesterone. It's quantified over here. So they were very responsive to progesterone. But importantly, when an inhibitor or blocking agent was used, so an inhibitor of rank ligand, this response could be abrogated. Now, denosumab is a monoclonal antibody that binds and neutralizes rank ligand, and it has a very good safety profile. It's widely used for osteoporosis and also for bone metastasis for many different types of cancer. So you can see here that this was very encouraging to see that this monoclonal antibody could abrogate progesterone responsiveness. So to test it as a prevention agent, it was absolutely necessary to turn to mouse models. And Emma used two different mouse models, but I'm going to show you one here. And this was very relevant because the BRCA1 p53 model expresses very high levels of rank expression on the surface of its cells, as you can see here. And so to establish a preclinical model, it was necessary to isolate, because we really wanted to recapitulate a prevention setting that would occur in a young adult woman. So precancerous cells were taken from these BRCA1 deficient p53 mice, and they were transplanted into a whole cohort of immunocompromised mice, and then they were randomized to receive either vehicle, or in this case it's called OPG, but it does the same thing. It binds and blocks the action of rank ligand. So they were randomized, and you can see here that there is a significant decrease in tumorigenesis. 2/3 of mice never developed tumours, whereas all the others did. So 2/3 of mice never developed tumours when they received the OPG blocking agent to block the activity of rank ligand. And when the other, I suppose, glands were assessed, there was no hyperplasia, so there was no hint of transformation. So this was a very important finding. And if we just put this together, so we've got these elevated levels of serum progesterone causing really, I'd say, an amplification of this pathway. So you've got more rank ligand signaling to these rank positive cells, which are expanded in number and have a predisposition to DNA damage, giving rise to a basal-like tumour. But the idea is to come in before any tumour develops, so to treat young women with a rank ligand inhibitor. In the case of humans, it will be called denosumab. And so this would abrogate this whole proliferative axis and prevent, it would actually switch off the proliferation of these cells. So because this is necessary for the proliferation of these cells. So the population should not expand and continue to accumulate DNA damage. And Jeff Linderman, who is also a breast oncologist, has actually set up a window study called BRCA-D, and these patients received 3 or 4 injections of denosumab, and the biopsies were collected at the pre-treatment stage and also the post-treatment stage, and they were compared. And these are the first 3 patients taken from this window study. And you can see in each case, if you compare pre-denosumab versus post-denosumab, there is a marked decrease in the number of these Ki-67 positive cells, so that's a marker of proliferation. So this is very encouraging. So for the last part of the talk, and it's only a very short section, so don't worry, I just want to finish on the patient-derived xenograft models, because we have spent the last 10 years deriving a whole bank of xenograft models to try and recapitulate the different subtypes of breast cancer and use them as preclinical models, because it's really, they are far superior to cell lines. So this work has been done mainly by Francois Vallon, and really the take-home message from this slide is that triple-negative or basal-like breast cancers engraft at the highest rate, and this is because they are the most aggressive. It's far more difficult to derive luminal subtypes because they have a better prognosis. Nevertheless, there are 2 subgroups here. There are the good prognosis and the poorer prognosis, and we were able to engraft the luminal Bs, which do carry a worse prognosis, and of course the HER2 positive. So we have the 3 major groups. And in very important proof of principle experiments, because we were at the Walter and Eliza Hall Institute, which has a very strong history in understanding survival molecules, we first looked at BCL2. And we did so because it is a key survival gene that keeps tumour cells alive. And BCL2 is actually a target in the estrogen pathway. So BCL2 is present in about 75% of breast cancers. And the idea here was to test these inhibitors in these patient-derived xenograft models and try and switch this balance so that the tumour cells would now be programmed to die by the process of apoptosis. So because if you can ablate the activity of this, that would be the end result. So in triple-negative breast cancers, we saw that this molecule ABT737, which neutralizes BCL2 plus 2 other members, it had very little effect, very disappointing. However, when it was combined with chemotherapy, docetaxel, you can see a very dramatic response. So there was a huge decrease in tumour growth and a huge increase in animal survival. So combination therapy was very effective. So that was triple-negative breast cancers. What about estrogen receptor positive cancers? Because these, of course, account for 75% of breast cancers. So here, again, we see an even more dramatic response. This really is baseline. There were no tumours palpable at all. So a combination therapy of tamoxifen and ABT737 completely melts these tumours. And we were able, there are different drugs now. So there's Abbott 737, you probably will have seen this in the news, and Abbott 199, which has just been, well, it's not, I don't think it's been okayed here, but certainly it's about to be in the US for chronic lymphoid leukemia. It's very, very effective. So through a comparison of these two drugs, we were able to show that it's actually BCL2 that is the important target molecule here, not the other members. And this has led to a clinical trial being led by G Linderman for advanced stage ER-positive breast cancers, and some of the results are coming through now, and they too look very encouraging. So I'll finish with really the most important slide, because this is the team without whom the work would not have been possible. So this is my very close and long-term collaborator, G Linderman. I've shown you the work today mainly of Anne Rios, Nyang Fu, Emma and Bhupinder, but they are all supported by an incredible team of people. I think there's Francois, he does all of the transplantation assays and has derived the PDX bank. And lastly, I would like to really acknowledge past members that really launched our work, these three members, and Gordon Smythe, he's a very close collaborator on all the bioinformatics, and Amgen, especially Bill Dougal. Of course, the donors, we would not have been able to do this work without them. Kay Confab for the familial breast cancers, the Royal Melbourne Tissue Bank, the Victorian Cancer Biobank, and all of our funders. Thank you for your attention.
Thank you very much for that excellent presentation. This is open for questions. Before I throw it open, maybe I can ask my own question, and that is, understandably, you focus on the stem cell compartment and the hierarchy of epithelial cells, but in the development of luminal structures elsewhere, the surrounding mesenchyme plays an important role. Does that influence the way that breast ducts and lobules develop?
Yes, I think the epithelial stromal interactions are very important, and they play a major role because they're responsible for the repertoire of cytokines and growth factors that ultimately drive this. But I think it's easier to do that in the context of tumours. So, altering the stromal environment and seeing if you can shut down tumour growth. So we are interested in that angle, but not in the normal developmental context. But they are key, as you point.
Yes, back there, please. Yes, please.
Well, we don't know the mechanism, but if you go back to that hierarchy, I think it's almost inevitable that it reflects the differentiation state. So I think that the cells that are proliferating with every menstrual cycle, throughout a lifetime, if they are never switched off, and go into a sort of dormant state, I think the risk of developing breast cancer is going to be slightly elevated. So pregnancy will, and especially a full pregnancy and early lactation, so they say that if you are 20 and have an early lactation, that is incredibly beneficial, because I think it's altering the state of the cells, the stem and the progenitor cells. It's driving them into a slightly more differentiated state, a more mature state, so they are less susceptible to becoming, well, cancerous, because I think it's a combination of proliferation and I suppose, luck in a sense, acquiring genetic mishaps. I mean, unless there's some underlying genetic predisposition. But if you have a very actively dividing cell, that cell is much more likely to acquire genetic mutations.
Great, thanks. Yes, next please.
Very interested in the cytological work and the BRCA1 patients. The BRCA2 seems to have gone by the board. They're all into BRCA1 now.
No, I think BRCA2 is still, it's a big question.
My question is, with this type of tools that you have for looking at precancerous or potentially cancerous cells, do you think there might be a role for looking at indolent cells in breast biopsy to determine which of the 1 in 8 patients is going to develop breast cancer? This is apart from the BRCA gene. Would it be valuable, do you think, to start looking not so much at mammography or MRI? Do you think there's a possibility that you could screen cytologically?
I think it's going to be difficult in the case of sporadic breast cancers, which are responsible for the majority. But in the case of familial, including BRCA2 and so-called BRCA-X, I think it would be a very valuable tool. Because we already see, so we see this aberrant growth behaviour with BRCA1. With BRCA2, although it's not clearly defined, you can read out aberrant activity, and BRCA-X is yet to be, fully investigated. So biopsies from these women may indicate, the presence of an aberrant cell type with aberrant growth properties. But I would never suggest it as a substitute for treatment, for example, with an agent such as denosumab, or for heightened surveillance, which these women have to undergo every year, or even a prophylactic double mastectomy, because the biopsy would only be from one area. You could never be sure what's happening in the other areas.
Great, yes please.
What percentage of breast cancers are caused by the BRCA mutation?
It's 1 in 400 in Australia, so it's about 2%. 1 in 400 women carry a BRCA1 mutation, and the proportion, so it's a 65% chance until the age of 70. In the US they say it's an 85% chance till the age of 70. I think it's closer to the 65%.
So in other words, there's a lot of gene mutations causing
Yes.
breast cancers that haven't been discovered.
Yes, that's true. These are the high penetrance genes. There are a lot of low penetrance genes that are yet to be identified, and they act together. So I think 1 will not be sufficient, but you need a mutation in 2 or 3, and that would account for the predisposition.
So, yes, next please.
I might have missed it, sorry if I did. When you used your brain inhibitor, what effect did it actually have on the mammary stem cell population, not just the luminal population? And also, have you used that with any other mouse models of breast cancer, just to see if it has an effect on the cancer in general, not just BRCA1 mutation?
So in answer to your first question, yes, we actually, I didn't put that in because in the mouse model we showed that rank inhibition compromised mammary stem cell activity. But in human breast tissue there is this difference. Remember, the mammary stem cell in human breast does not express rank. It's only the luminal progenitor population. So does it have wider activity? Yes, in the Wnt model we saw activity, perhaps not as pronounced as the BRCA1 p53.
3 last questions, and first the gentleman there. Okay.
Yeah, so there's a pilot study going on at the moment, but by the end of next year, we hope that there will be an international trial. There should be, so it will involve actually an Austrian team and G Linderman. That is all, the protocols and everything are all underway. So we're not going to know probably for 10 years because you need thousands of women. So as I mentioned briefly that it has a good safety profile, but the dose has to be correct. And, you know, before it is approved, you have to be certain. So this they are probably going to enroll, I think, 3,000 women. They're all, you know, all BRCA1 mutation carriers, and this can only be done on an international level. It will also give very important information about ovarian cancer. So it will. The idea is to prevent both breast and ovarian.
Yes, please.
Hi, Jack. You spoke about the cellular codependency of the mammary gland, where Aberrant simulate positive cells, basal cells, positive cells. But when we talk about the kind of cancer cell origin concept, you tend to see the idea of homogeneous cell population. So my question would be, to what extent is there a persistence of this kind of codependency on different subsets of cells that might suggest maybe inducing differentiation using things like progesterone might be an appropriate approach to treating breast cancer?
They're good questions and difficult to answer at this stage because, of course, first of all, one needs to understand the hierarchy, but keep in mind that we're not looking at single cells. They're all very precisely arranged in ducts. And I don't think there is a lot of plasticity from current work that's coming through, but yeah, I don't think it's necessarily going to be unidirectional, but we don't truly understand the configuration. So we know we've got a luminal layer and a basal layer, but we don't understand the precise configuration of cells along the ducts at the branch points near the nipple region versus the distal region where the breast cancers arise. There are a lot of questions. I'm sorry, I can't answer that one.
One last question, thank you.
Yeah, I guess along that same line with the rank ligand inhibitors, I know you mentioned that that's important in bone. So why is it so safe? Wouldn't there be long-term effects on bone health of taking rank ligand inhibitors as a long-term prevention?
Well, for osteoporosis, it's a low dose. It's long-term use of a low dose, and it's very safe. Once you start to increase the dose, though, there is one trial that has used very high levels, I'd say industrial doses. There are potential side effects. They're still rare, but the side effects are not good, you know, osteonecrosis of the jaw. So the dosing has to be quick.
So I'm sorry, I think we'll need to call it a day there. Just look, I think you'll all agree with me, a fabulous presentation, a very elegant studies. I'm a bit of a morphologist myself, but the 3D stuff that you showed at the beginning just blows me away. It really is fantastic stuff, together with very elegant studies around the cell lineage tracking and the animal models. And I think to me one of the important bits that you've underlined today is how important it is to understand physiological mechanisms and in development in unpacking the mysteries of carcinogenesis. Those, thank you very much. Please join me in thanking. So that draws to a close the proceedings, and I wish you all safe home. Thank you.